students, good morning and welcome back to biology 2. Okay, so we are now going to chapter 5, population genetics. Okay, so what is population genetics? It is a continuation from the previous chapter. This time it is about the genetic variation within the population. So population is subject to four main evolutionary processes: natural selection, genetic drift, mutation, and gene flow. So this in the branch of biology examines the phenomena as adaptation and speciation. So as you can see in this comic, different genes would have different afflictions. So for the first panel, so the first patient I'm seeing today has an alpha thalassemia, might be Asian. Then someone with sickle cell anemia, wow, almost definitely of African descent. And here's a middle-aged black guy, should probably look for hypertension. A patient diagnosed with Tay-Sachs disease, I bet they are an Ashkenazi Jew. Okay, so meaning that different type of ethnics actually have different type of common diseases. So within each of these populations, there are different common diseases. So gene pool. The gene pool will be the total genes of everybody in that population. Okay, so all species have a shared gene pool, and members of species can interbreed and produce fertile offspring. Populations. Populations are a group of the same species living in an area. No two individuals are exactly alike, so they have variations. One of which individuals survive and pass on their traits. So modern synthesis theory. So modern synthesis theory, in 1940s, you have the most comprehensive theory of evolution, which is called the modern synthesis theory. And this theory combines Darwinian selection and Mendelian inheritance. So the current theory on evolution was introduced by Fisher and Wright. Until then, many did not accept that Darwin's theory of natural selection could drive evolution. Okay, so in MST, it recognizes that genes are responsible for the inheritance of characteristics, and recognizes that populations, not individuals, evolve. And it also recognizes that more forces contribute to evolution and not just natural selection. And it recognizes that speciation usually is due to gradual accumulation of small genetic changes. Hardy-Weinberg principle. So Hardy-Weinberg principle, it is made by two people, G.H. Hardy, which is a mathematician, and Wilhelm Weinberg, who is a German physician and obstetrician gynecologist. So the Hardy-Weinberg principle is used to describe a non-evolving population, and it is also showing that the shuffling of alleles by meiosis and random fertilization have no effect on the overall gene pool. Natural populations are not expected to actually be in a Hardy-Weinberg equilibrium. So the principle says the allele and genotype frequencies in a population remain constant from generation to generation in the absence of other evolutionary influences. So the deviation from Hardy-Weinberg equilibrium usually results in evolution. Understanding a non-evolving population helps us to understand how evolution occurs. 5 assumptions of the Hardy-Weinberg principle. Number 1. Large population size. So populations have fluctuation in allele frequencies. Number 2. No gene flow. Migration can change the frequency of an allele by bringing in new alleles to the population. Number 3. No net mutations. If alleles change from one to another, this will change the frequencies of those alleles. Number 4. Random mating. If certain traits are more desirable, the individuals with those traits will be selected, and this will not allow for random mixings of alleles. Number 5. No natural selection. If certain individuals survive and reproduce at a higher rate than others, then the offspring will carry those genes and the frequency will change for the next generation. So, in short, the gene pool of a non-evolving population remains constant over multiple generations. So, allele frequency does not change. Hardy-Weinberg equation goes with 1.0 equals to p squared plus 2pq plus q squared, where p squared is the frequency of AA genotype, where p squared is frequency of capital A genotype, big A. 2pq is frequency of big A small a, which is heterozygous, and q squared is frequency of small a genotype. So to determine allele frequency using Hardy-Weinberg, it is 1.0 equals to p plus q. where P is frequency of big ALL and Q is frequency of small ALL. So in this picture, you can see that there is a parent population. Okay, there are phenotypes which are red and white. So genotypes for this red and white, okay, you have three genotypes. You have the first genotype is the big R, okay, two dominants. Second will be big R, small r, one dominant, one recessive. And the third one is small r, which is both recessive, which is white color. So let's say that in the number of plants, which is total 500, 220 are big R, and 160 is big R, small r, and the rest is small r. In that, the most will be the double dominant. So in terms of genotype frequencies, how many do we have? 320 times 2 because there are two big R alleles over 100. So you get 0.64. And therefore, big R, small r, you have 160, so it is 0.32. And the small r is 0.04, which is 20 over 500. So we count the number of alleles. Because the number of alleles for big R is 640, it is due to 320 flowers times 2. Okay. What about big R, small r? Big R, small r have 160 big R and 160 small r. While the one with white color, small r, has 40. Why? Because there are 20 flowers with 2 of the alleles in each flower. So there are 40. If you combine them together, you get 800, which is the allele for red, and 200 is the allele for white. So when you combine them together, they actually total to 1. So 800 over 1000 is 0.8, 200 over 1000 is 0.2. So when they are plus together, they are 1.0. Okay, so for the next generation, if you combine the gametes from the first generation of parents, you can actually calculate how much is the probability for the next generation. So from the number that we have found in the previous page, P squared is 0.64, 2PQ is 0.32, and Q squared is 0.04. According to this Punnett square, the big R is 0.8, the small r is 0.2. If there are two big R's together, 0.8 times 0.8 is 0.64. If the big R, small r mated together, you get 0.16 plus 0.16, it is 0.32. And 0.2 times 0.2, it is 0.04. Can you see the numbers? The numbers is exactly the same as what we have calculated in the previous page. Okay, so whatever happens, the number of the LL, okay, the frequencies are constant. So macroevolution of species. So it is change in allele frequencies that occur over time within a population. The change is due to 4 different processes: mutation, natural selection, gene flow, and genetic drift. And mutation, macroevolution over time may lead to speciation of an appearance of a novel structure, which is a new characteristic, okay, that we also call macroevolution. So macro and microevolution describe fundamentally identical process on different scales. So macroevolutions happen little by little. Macro, there's a lot of change. Yeah. Causes of macroevolution. First is genetic drift. So it is a small change of the gene pool by chance. Second will be natural selection. It is successive reproduction based on heritable traits result in selected alleles being passed down to relatively more offspring. Natural selection is successive reproduction based on heritable traits result in selected alleles being passed to relatively more offspring. Okay, so it's called Darwinian inheritance. So if they are able to withstand the changing environment, they get to live. And when they live, they can breed. And when they breed, they have children who carry that trait that the parents have. Gene flow. It is a genetic exchange due to the migration of fertile individuals or gametes between population. If you can see here, population A, population B. Okay, both of these they have gene flow because some of these individuals go from A to B to breed. Some of them go from B to A to breed. So the gene from A and B can be found in both populations. So causes of microevolution. First of all is mutation. Mutation is a change in organism DNA. Mutations can be transmitted in gametes to offspring. So, factors that causes genetic drift. First of all is the bottleneck effect. Bottleneck effect is a drastic reduction in population. For example, due to a disaster like volcanoes, earthquakes, and landslides, it causes the reduced genetic variation. So population may not be able to adapt to the new selection pressures. Founder effect occurs when a new colony is started by few members of the original population. It leads to reduced genetic variation and may lead to speciation. You can see this picture. Okay, you can see there's a lot of marble in the bottle. You have a lot of yellows, a lot of blues, and some reds. Okay, so let's say that in the bottleneck effect, only few will survive, right? Can you see how narrow the neck of the bottle is compared to the body? Okay, this means that through a certain disaster or a calamity, only few can escape the bottle to survive, meanwhile others are trapped inside. Okay. In this simulation, we can see that there's a drastic reduction in the population. Okay, instead of having a lot of yellows, you have a lot of blues and some yellows and no reds at all. So you can say that the red did not survive. So the surviving individuals will be making the next generation. So in the next generation, the population totally has shifted to many of them are blue and some yellow and no reds because these are the survivors. Loss of genetic variation. Cheetahs have very little genetic variation in their gene pool. This can probably be attributed to a population bottleneck that they experienced around 10,000 years ago. So they barely avoid extinction at the end of the last ice age. So here is a founder's effect. You can find that there's a lot of ladybirds, some of them red, some of them yellow. But one of the reds go to another island and they make a population there. So in the end, you have an island fully with red ladybirds. Heterozygote advantage. So heterozygote advantage, they favor heterozygotes, VA small a. So the thing is, for heterozygote advantage, both VA small a are maintained, but it doesn't actually remove less susceptible left from the population. Okay, for example, the real life case of sickle cell anemia. So in sickle cell anemia, you are born with a certain lack of blood due to the red blood cells shaped like a sickle. Okay, so this sickle shaped cell cannot bring oxygen as good as the regular red blood cell, which is round in shape. Okay, so if you look at it, it is kind of like a disadvantage. You cannot carry a lot of oxygen, you get tired easily. But the thing is, in a certain area in Africa, this is actually an advantage for them to survive malaria. So malaria is a blood parasite. So the parasite lives in your red blood cell. If somebody actually have this sickle cell trait, they're able to prevent themselves from being infected. Okay, right. Compared to regular people with regular red blood cells, the round red blood cells, this makes it even more susceptible to infection of this malaria parasite. Okay, so in this case, normal people will die of malaria. People with sickle cell trait, they live, they survive. But in other case, people born with sickle cell disease mean that all of their cells are sickle shaped. There's no normals. They actually die because they do not have any oxygen and their blood actually have clotting. So who survives? The heterozygotes. Modes of natural selection. Okay. In the modes of natural selection, the first is the directional selection. Directional selection would favor individuals at one end of the phenotypic range. So it is most common during times of environmental change or when moving to new habitats. If you can see this picture, okay, 1977, drier, there's a big depth, and then it goes low again. 1980, drier, goes high again, and then goes low back. Okay. And then in 1982, it goes dry, and then you can see that there's pattern here. Whenever it's dry, the big depth increases compared to the wet year. So in dry season, 1977 in Daphne Major, okay, plants dried up, only seeds are left. So median and ground finches can select two types of seeds: small spurge seeds, which is on the left, and hard woody caltrop seeds, which are on the right. Okay, they are a little bit they're a little bit pointy, a little bit sharp. It's really hard to eat. but you know there's a food inside of that. For cracking open caltrop seed, big finches with 11 mm deep need 10 seconds only. Medium birds 10.5 meters deep to 15 seconds. Small birds, such as the small ground finch, cactus finch, they take too long to crack. Many of them give up. So instead, the small big ones can only manage to eat spurge seeds. Okay. In 1977, there's a major drought. No small spurge seeds are left, leaving only the caltrop. And this actually causes many birds to die because they cannot crack open the big caltrop seeds. After a few years, the finch population recovered. And what's left? You can see that during that time, a lot of the birds that are surviving, they have big beaks. Okay, now 9.2 mm or average depth. Okay, and the beak size is 8 to 11 mm. Okay, mean that after that is like much more larger, 50% bigger. So what happens? You can say that due to the drought, the weak one, the small ones, are unable to feed themselves and they die, leaving only the big ones. So, conclusion. Population has shifted because the finches with bigger beaks have better survival chance. Survival will produce a bigger fraction of big beaks in the next generation. So, deciding selection. It favors extreme over intermediate phenotypes, and it occurs when environmental change favors an extreme phenotype. So, this is a Florida oilfield mice. Okay, the body is brown on the top and has a white underbelly. So there are many predators for this mouse: fox, weasel, owls, and cats. In natural woody ground soil habitat, it is well camouflaged. You know, you can see that the ground is brown and the mouse is brown, so it is easily hidden. But occasionally, a few mice with lighter colored coats are born due to accidental mutation. So the sea levels rose dramatically 6,000 years ago, turning the coastal habitat to barrier islands. Both light and dark furred mice are stranded in the islands. Barrier islands have white sands, and the minority light fur mice has greater survival because they have better camouflage on the sand, unlike the dark ones. So the survivors reproduce and pass down the light pigmentation gene to the offspring. So as the original oilfield mice survives in their own ground habitat, it favors the brown camouflage rather than light pigmentation. So the ones in the intermediate pigment color, light brown, stands out in both habitats, you know, the mainland and also the island. So it does not survive. So the only way to survive is either you are brown or you are light colored. So there are two extremes, dark and light. There's no grays. Stabilizing selection. This favors intermediate over extreme phenotypes. So this reduces variation and maintains current average. For example, the human birth weight. For babies with low birth weight, they lose heat more quickly and get ill from infectious disease more easily. Babies of large body weight, much more difficult to deliver through the pelvis. Infants of a more medium weight survive much more often. And for the larger or small babies, the baby mortality is much higher. Summary, you can see that in the beginning, you can see there's original population of phenotypes of these mice. If you have directional selection, only one type of fur color or fur tone is preferred by the environment. Okay. Diversifying means that there's no middle, they are only the extremes, the white or the dark. So Bayesian selection, we select the medium of it. Alright, that's the end of the chapter 5. Thank you and see you in the next chapter.